All right, good evening, everyone. My name is Hunter, and this is content review for Principles of Biology students. Today we are doing membrane potential. And I want to start off this week with a little bit of encouragement. We just had exams. It's the eighth week of a 10-week term. We've got more exams coming up. Eighth week is when I feel the roughest. So I just want to affirm for folks that it's okay if it feels hard. It's okay to take care of yourself. Jumping into today's topic, it's kind of big. And I remember being really scared of this one. You do not have to be. But if you can spend some time with this and really start to understand the different moving parts and um, really get acquainted with these models, I'll try and give some key insights that were helpful for me in unlocking this topic because understanding this will make future topics so much more approachable. This keeps coming back up. We're doing echocardiograms right now. And because I remember because I spent the time with this, um, the understanding is coming a lot easier. I feel like understanding this unlocks a lot of other topics. So we're going to start with an example, and then we will kind of backtrack into why these things work. All right. So how do ion concentrations affect neuron function? So much of our early understanding of neuron function was based on studies of the squid's giant axon. And squids move through the water by contracting the muscles of the mantle, and this movement compresses water inside the mantle and forces it out the siphon. A squid can change the direction of movement by directing the flow from the siphon either toward a forward or backward relative to its head or anterior end. And so I'm going to label a couple of things because this got confusing. This is anterior, and we may also call this the tail later, and this is the head. The mantle's already labeled, and we also have a ganglion labeled here. So in which direction would the squid move if water flow from the siphon were directed toward the head end of the organism? So if water from the mantle is directed this way, presumably we're in some kind of aqueous medium, right? The ocean, perhaps. And so if we jet water that direction, it's going to create a force, a restoring force in the opposite direction. And so movement would be opposite. So the squid would move backward or towards the tail end. Conversely, if the siphon were directed backward so that it was pointed towards the mantle, if it was pointed that way, movement would be towards the head. Yeah, I don't think I need to rewrite that. Um, okay, the cropping has chopped off my numbers, but we're still good. When squid are startled or in danger, they can simultaneously contract all muscles of the mantle to forcefully jet water out of the siphon and escape. And escape rapidly. Assume the mantle of the squid is 30 centimeters in length, which is about 12 inches, and the distance from the brain to the mantle opening is another 8 centimeters, about 3 inches. For all muscles of the mantle to contract simultaneously, a nerve signal sent from the brain must reach all parts of the mantle at the same time, even though they are different distances from that space. So on the diagram of the squid above, draw 3 neurons and assume all of them originate from the ganglion indicated, and the ganglion is stimulated by action of a single motor neuron from the brain. Neuron 1 innervates the mantle, muscles nearest the brain. Neuron 2 innervates the muscles in the mid region of the mantle, and neuron 3 innervates the muscles at the tail end of the mantle. I'm going to draw these in red. We're going to start at our ganglion. And we have neuron 1 was the near end, so it's right here. And then we had neuron 2 was kind of the mid region. So that's 1, 2. And then neuron 3 went all the way to the tail over here. Okay, so we have our different lengths. In invertebrates, like the squid, how must the nervous system design be modified to allow both muscles nearest the brain and those furthest from it to contract simultaneously? Modify your drawing to indicate any necessary changes in the size or structure of the three neurons that would be required and explain your reasoning. So the first most important thing that you need to know about this is that invertebrates don't have myelinated nerves. Myelin, you will remember, is a insulating sheath around a nerve fiber that speeds up signal conduction. So without that, we have to use some other mechanism of speeding it up or slowing it down so that everything fires at the same time. And the way that we do this is by simply increasing the size of the neuron, increasing the diameter for a faster action potential. So redrawing this, I'm just going to use a thicker marker. Axon 2 is a little bit longer, or wider, rather. And axon 3 is thicker still. Cool. If you're taking physics in your electrophysics portion, you will actually have an opportunity to examine the mathematical basis for why this happens. But the easy sort of mental shortcut here is bigger pipes, faster current, right? The current here, we're talking about an electrical signal rather than the flow of water. Um, Fluid dynamics get a little bit trickier, but I don't want to distract too much from the analogy here. Bigger pipe, faster travel of current, of electrical current. All right. Cool. Researchers discovered that they could remove these giant axons from the squid, and with some skill, these axons could be maintained outside of the body if held in ion solutions of the same concentration as the squid's extracellular fluids. If stimulated with a microelectrode, these isolated axons would generate action potentials. By recording the potential differences between an electrode on the axon versus one of the fluid bathing the axon, researchers could also record the potential difference at rest and any change in potential difference that occurred as a nerve impulse or action potential was generated. Basically, this example is talking about the basis for how we discovered the differences in these ion concentrations and that this is the basis for how our nervous system works. And I really, really like that this worksheet dives into this. So the ion concentrations of the the ion concentrations the researchers recorded for the squid neuron are listed in the following table. And again, just a little science literacy, ion concentrations in millimolar for intra, inside, and extra, outside cellular fluids of the squid axon. We have our different ions here. The ones that we are going to be paying the most attention to for this live stream, at least, and probably in your class, are potassium and sodium. You'll notice that potassium is very high relative, it's very high on the inside relative to the outside, while sodium is very high on the outside. So I'm going to draw a cell here. So we have high potassium on the inside and high sodium on the outside. Some of the concentrations for a human are listed below. Again, you will notice the same trend. Potassium is high on the inside and low. Potassium is low on the outside. And then we're given this equation. And this equation is helpful for quantitatively explaining or measuring what we're about to show you, basically. Using the Nernst equation, calculate the equilibrium potential for potassium and sodium in the human. All right, we're going to run right through this. So for potassium, 58 millivolts over Z is our ion valence. Sodium and potassium each have a valence of 1 times log. The ion concentration of potassium outside is 5 to 140 inside. And if you plug that into your calculator, you're going to get negative 84 millivolts. And this is for potassium. For sodium, it's very similar, except it's inverse. We have 150 in our numerator, that's the outside concentration, and 15, 58 millivolts. And this is the equilibrium potential. So this begs some questions, which we're going to get into. Looking at the squid, you can just plug and chug in this equation. I'm just going to give you the answers right now. For potassium, you have negative 75.5 millivolts. And for sodium, we have positive 54.8 millivolts. You will notice that these values are fairly similar. And what this tells us is that the neurons behave very similarly across species. So values in this range are typical for most neurons. The difference in actual concentrations of the ions, human system versus squid system, can be accounted for because the squid is a marine organism and the sodium potassium concentrations in the ocean are playing a factor here in the excitability of these membranes. So in a typical neuron, which ion has the greatest influence on the membrane potential at rest? In other words, flux of which ion contributes the most to the resting membrane potential, and explain why this occurs. I'm just going to write this number here. Negative 70 millivolts is around the resting membrane potential for humans, and this is an important number that I would have memorized for the purposes of your exams. At rest, all sodium voltage gated channels are closed. And this is where I wish this worksheet was organized a little bit differently. But in our axon, we have these cells which regulate the movement of sodium and potassium. And the reason that we have this is we are moving them against their concentration gradients. Remember, we were talking about osmolarity, and this is true for ions as well, where they always want to move from areas of high concentration to areas of low concentration. So left to its own devices, sodium would want to move into the cell, and potassium would want to move out to equalize their concentrations. But if we do that, then we have a net neutral membrane with no difference across it, and this behavior would not be possible. And so thanks to sodium potassium ATPase, we can move them against their concentration gradients and maintain this gradient. Flux of which ion contributes the most? It is important to know that the cell is more permeable, the cell membrane is more permeable to potassium than it is to sodium. So outward flow of potassium just doing its thing is counterbalanced by electrical potential difference resulting from not just potassium gradients, but also other anions and cations. Sodium channels are closed at rest, so not contributing to the resting membrane potential as much. Right? In calculating the resting and action potentials of the axon, we generally don't worry about the concentration differences of calcium or chloride ions. Explain why this is so. Simply put, neuron membranes are not permeable to calcium or chloride. What effect would the following changes in extracellular potassium concentrations be likely to have on the resting membrane potentials of neurons of a human? So if we increase extracellular potassium from 5 millimolar to if we decrease from 5 millimolar to 2 millimolar. So remember that in a normal nerve cell, we had high potassium on the inside and high sodium on the outside. Salty banana is the mnemonic here. Your cell is the banana, which is high in potassium. Salt is sprinkled outside. So that's in the extracellular fluid. If we further decreased the already low concentration of potassium outside of the cell, what is that going to do to the potential difference? So the voltage potential just describes a difference in charge from one side of a membrane to another. If we further decrease that, that difference is going to get even bigger, and that should be tickling some nerve bundles in your brain. Hyperpolarizing, more polar. So this is going to hyperpolarizes the membrane. Is this going to have an excitatory or an inhibitory effect? To answer that question, we have to know that depolarizing is what happens when an action potential is propagated. That's an action potential being the signal down the axon. So hyperpolarizing inhibits. So lowering the potassium concentration outside is going to inhibit or reduce the likelihood that an action potential will occur. Inhibition. And you can't really read it on here, but I will just read it off my other sheet here. What if we changed the extracellular potassium from 5 millimolar to 20 millimolar? So this is kind of the opposite, where now we are raising the potassium to meet the sodium, and we still have high potassium inside the cell. You can't see that. We can actually use our Nernst equation to find out what this difference is going to be quantitatively. And we would end up with a value for potassium that is at negative 49 millivolts. Up here it was negative 107 millivolts. So really, really, really big difference. Negative 45 49 millivolts is really close to the threshold potential for most organisms. And what that means is you would have constant firing of the nerve. In this kind of situation, you might have tetanic contractions, so spasms. If this is in cardiac muscle or respiratory muscle, this can lead to death very quickly. If there's no cessation. Um, okay. How do neurons function to transmit information? And here we get to create a model. I'm going to freestyle for a little bit and just kind of talk about some of the elements that are important here, some of the key insights that really helped me. So this is our nerve cell in cross section. Generally, it has a negative charge on the inside, which you can remember by thinking about proteins, the backbones of DNA. All of these things have a net negative charge to them. So the inside of the cell is usually a little bit a little bit negative. And then you have an accumulation of positive charge on the outside. Remember that that's where all the sodium is hanging out. And on the inside is where we have potassium hanging out. To maintain that difference, we have sodium potassium ATPase, which is pushing potassium, this is sodium, 3 sodium out and 2 potassium in against their concentration gradients to maintain this difference in potential. So we have potassium, sodium, negative organic ions we talked about. We have sodium potassium ATPase. Also on here, we have leak channels where you're going to get a little bit of passive diffusion of potassium out of the cell. And then you have, I'm going to do these in a different color, voltage gated sodium channels. These are closed normally at rest. When an action potential arrives at threshold, they will snap open, allowing sodium to rush into the cell. That further depolarizes the membrane, which pushes an action potential down. More about that in a second. There's our voltage-gated ion channels, sodium gates. We also have potassium gates, gated potassium channels, which are closed at rest. And when they open, potassium rushes out. Excuse me. Voltage-gated sodium channels and voltage-gated potassium channels open about the same time, but they close at different rates. And the closing at different rates is part of what is responsible for the refractory period, which is basically the recovery state of the action potential. So it hits threshold, fires, resets with the potassium rushing back in, and it slowly closes until you're hyperpolarized, right? It's what allows for that hyperpolarization. And then it gets back to resting potential. There's your rest. You can't see that. Come on. There's your peak action potential. Cool. That's also a good one to be able to draw from memory. Let's draw an axon. So we have our cell body with our nucleus. It has dendrites, which can interface with different tissues, with other nerve cells. And then we have the axon. And right here is a little bit thicker part called the axon hillock. The axon hillock is where an action potential is generated. Once it hits threshold there, sodium channels open along the length of that axon, pushing that wave of depolarization down towards the next cell. So in this model here, the one on the left would be the pre-synaptic neuron, and the one on the right would be the post-synaptic neuron. Drawing it this way is kind of helpful for me. We'll come back to that in a second. Pre and post. The space in between them is your synaptic cleft, and we can draw a much bigger version of that now. So if we have pre and post-synaptic cells, when our action potential arrives at a synapse, a junction between two nerve cells. How do I want to draw this? Action potential arrives at the cell. Calcium is allowed to flood into the presynaptic cell, which stimulates the formation or the exocytosis of vesicles containing neurotransmitter. So exocytosis means bubble moves to the cell membrane and it releases neurotransmitter into the synaptic cleft. If the postsynaptic neuron has a receptor for that neurotransmitter, and that those neurotransmitter can have inhibitory effect or excitatory effects. If it has a receptor for it, that neurotransmitter will take action at the synapse. Well, I'm going to go really far down a rabbit hole right now, and we're just building a model here for thinking about this. So synaptic vesicles carry neurotransmitters. We already kind of talked about the axon, dendrites, and cell bodies. We talked about calcium. Calcium are an important signaling molecule for other proteins inside presynaptic nerve cells to release these neurotransmitters. Sodium gates we talked about, potassium gates we talked about. Sodium and potassium gates open at the same time, which equalizes charge on either side of that membrane. It depolarizes it, right? And then the sodium gates have two gates. One snaps shut immediately as soon as it gets to peak, and the other one closes a little bit slower, and that's part of the refractory period. Potassium gates close the slowest, and those are, they're moving they're moving a lot slower in general, opening and closing. And that difference in timing is what accounts for the hyperpolarization that I drew here below the resting threshold. That's why it kind of has to correct after they fully close. Okay. Salty banana, sodium potassium ATPase, sodium potassium pumps maintain that electrochemical gradient. There's some leakiness for both potassium and sodium, but more so for potassium. And sodium and potassium voltage regulated gates are responsible for depolarizing and propagation of the signal for sodium. And for potassium, it's for hyperpolarizing the cell and resetting it back to resting potential. That's this whole worksheet summarized right there. All right. Use your understanding of how action potentials are generated and propagated to answer the following questions. All cells maintain an ionic and therefore electrical potential difference across their membranes. In most cells, the potential difference is between -50 and -100 millivolts. That is, the inside of the cell is more negative than the outside by 50 to 100 millivolts. Although all cells in the body maintain this potential difference across their membranes, only certain cells, for example neurons, are capable of generating action potentials. How is this potential difference across the cell membrane generated? And this is sodium potassium pumps, or sodium potassium ATPase, if you want to get really specific. And they pump, I drew it up here, 3 sodium out, 2 potassium in. Okay. Yep. And that difference of 2 to 3 is what accounts for the general negative charge on the inside of the cell and the general positive outside of the cell. What characteristics of membranes allow cells to concentrate or exclude ions? Selective permeability should be a very familiar term by now. Ions such as sodium and potassium cannot diffuse across the phospholipid bilayer. They require specific channels. Specific channels or active transport. What is it about neurons or nerve cells that make their properties different from those of other cells? In other words, what enable nerve cells to produce action potentials? So all cells generate a potential difference across their membranes. However, only nerve cells have voltage-gated ion channels. Only nerve cells, or excitable membranes rather, more specifically, have voltage-gated ion channels. Voltage gated just means that when it hits a specific voltage or potential difference, the gates snap open. As they reach whatever peak or depolarization, they close. That's all that means. Postsynaptic nerve cells also have chemically gated ion channels. Chemically, as opposed to voltage gated. And these respond to the neurotransmitters that I mentioned a moment ago. While we're here, um, that's usually lowercase. Acetylcholine is one that you're going to see a lot. Serotonin is a neurotransmitter that a lot of people have heard of. Dopamine is another one. These can have inhibitory or excitatory effects depending on the synapse that they are in. Acetylcholine almost always, usually, excitatory, especially if we're talking about neuromuscular junctions. That may or may not be relevant for you guys yet, but it's a good one to know. How is an action potential started and propagated? This is a good one to go back to your textbook and actually like look at all of the diagrams that they give you. Figure 43.7 in particular, in your Biological Sciences 7th edition will give you a good breakdown of this. And I have to think about how I want to draw this for a second. Yeah. Okay. So when we have our nerve cell and its dendrites, axon, down to the next nerve cell, we have a local depolarization, which usually happens at these dendrites, right? Summates the depolarization, which starts an action potential at the axon hillock. So local depolarization of the neuron membrane open some sodium gates. How do I want? Yeah, okay. So in red, I'm kind of drawing our little bit of depolarization, these signals kind of accumulating. And at the axon hillock, once we hit threshold potential of about negative 55 millivolts, we get an action potential. And that depolarization is opening sodium channels. So sodium is allowed to move into the nerve cell membrane. Once we hit negative 55, yeah, once we hit negative 55 millivolts, the next one snaps open. Jeez. The next one snaps open. Sodium's able to move in. We hit threshold. The next one opens up. Sodium moves in. We hit threshold. So that's that wave of depolarization that we're talking about. When the potential difference across the membrane at this site on the axon, the axon hillock, excuse me, at any point, when the when we depolarize sufficiently, when we hit peak depolarization at positive 30 millivolts, now things start to close. So we go from open, we start to close these. And at that time, potassium channels open. And this is going to get us back to resting membrane potential. Yep, until this point, potassium channels were closed. Once we hit peak action potential, potassium channels open, and we start to repolarize. So up here we were at negative 55 millivolts. We hit peak depolarization at positive 30 millivolts. Those potassium channels open. So now the potassium channels are open, we're repolarizing the cell. And once we get back to negative 70 millivolts, which was resting, those potassium channels start to close again, but they close slowly. So as they're closing slowly, potassium moves in nice and slow, still leaking in. So we get a little bit past resting potential and hyperpolarize. This is the refractory period. Because while it's hyperpolarized, we can't generate another action potential, and all of these gates won't respond to the next one if it were to arrive early. This limits the number of action potentials that can be generated per unit time. The refractory period also helps explain why action potentials only travel in one direction. So the refractory period is important for one-way action potential propagation and limits number of action potentials per unit time. Does generating an action potential require any direct or indirect energy input? Short answer is no. No energy required. It's all sort of, with the exception of sodium potassium ATPase, all of this is happening just as a result of the nascent, um, the latent electrochemical gradients. Yeah, so energy is only required indirectly for maintaining that resting membrane potential. What happens in time and space along the axon once an action potential begins? We've described this a couple different ways, right? Um, Earlier we were looking at what happens at a single site on the axon as it was depolarized. So the way I drew this, okay, you can see that. The way I drew this, we're meant to be looking at the same spot across time. And now we're talking about what happens along the whole length of the axon. So at the height of the action potential, that positive 30 millivolts, the inside of the axon is now more positive than the outside. More positively charged than outside. At least for that infinitesimally small moment in time. The sodium ions that enter the axon at this site diffuse in all directions from their point of entry. And as they diffuse, they change the potential difference across the membrane. The next site on the axon reaches threshold potential, and its sodium gates open. So I did kind of talk about that. So arranged in space, when this one hits threshold, the sodium diffuses up to the next one, which depolarizes it, which allows more sodium to come in. Those diffuse, depolarizes the cell, opens the gate, more sodium rushes in. And that's that linear propagation. Although the sodium ions diffuse in all directions from their point of entry, um, the action potential is generated only in one direction, again, because of that refractory period of these channel proteins. They have those two gates. There's the opening gate and then the closing gate. The closing gate closes more slowly, but that is keeping them from going backwards. What factors ultimately limit the ability of a system to respond, that is, continue to generate impulses? Um, Sodium potassium pumps ability will limit this. So let's and they're limiting this simply because they take time to establish resting potential. In experimental situations, isolated neurons are used to generate and record action potentials. Um, when the sodium potassium pumps in the neuron membrane are poisoned, if they're rendered non-functional, the axon is still able to generate action potentials in some cases. In some cases up to several hundred of them. But this occurs because the number of sodium and potassium ions that move across the membrane during an action potential is very small. Um, but on the other hand, the sodium and potassium ion concentrations inside and outside the neurons are measured in millimoles. If a molar solution of potassium ions contains 6.02 times 10 to the 23rd potassium ions per liter, then a millimolar solution contains 6.02 times 10 to the 20 uh, potassium ions per liter. And as a result, many action potentials can be generated before the potential difference across the membranes is reduced enough to prevent their generation. So they'll still work, but not forever. If an axon is stimulated in the middle of its length, nerve signals or action potentials will move out from the point of stimulus in both directions. Normally, however, nerve signals move in only one direction along neurons. Why is this? And this again is that refractory period. Of voltage gated sodium channels, voltage gated sodium channels. Um, Refractory period of voltage gated sodium channels is responsible for one-way propagation of a signal. It's coming out from the middle because that's where it was stimulated artificially. It's just going to go out from there. If it's going back towards a presynaptic cell, it's going to stop. It won't be able to go anywhere. If it goes down to the next postsynaptic cell, it may continue on to the next one if it has sufficient. Momentum isn't exactly the right word, but yeah, artificially no refractory period exists when action potentials are generated in both directions. Whether or not an action potential is generated in a postsynaptic neuron depends on a number of factors. What are these factors? What ultimately determines whether an action potential is generated in the postsynaptic neuron? This depends on a couple of things. We've got the type of neurotransmitter. That is it excitatory or inhibitory? We have the number or frequency of excitatory versus inhibitory neurotransmitters. And we have how close, so proximity, to the axon hillock. If the combination of all of these inputs depolarizes the postsynaptic neuron to threshold level, an action potential is generated. You'll notice we're not writing numbers down for a lot of these threshold levels, and that is because they are different for different kinds of excitable membranes. Um, for most neurons, especially the neuromuscular ones that you will be getting acquainted with, it's negative 55 millivolts. But that is not universally true. Diffusion is efficient over only very short distances. In fact, as you can see in the table, diffusion is efficient only for a distance of about 1 to 100 micrometers. How wide is a synapse? It's about 10 to 20 micrometers. Very, very, very tiny. So we're looking at 50 to 100 milliseconds um, for diffusion to take place across that distance. If a synapse were 2 times as wide, what effect would it have on the transmission of nerve signals from one neuron to the next? And how would this change affect the response time of an organism? The synapse were 2 times as wide. The time for diffusion would increase by the square, so 4 times. So I guess it's diameter squared. So if you have 2d squared, that's 4 times. Okay. If you examine neuron transmission within an organism, you will discover that every action potential generated is stereotyped. For example, every action potential reaches the same. Am I looking at the wrong one? No. For example, every action potential reaches the same maximum height and the same minimum height. In addition, the generation of action potentials is an all or none phenomenon. That is, once the potential difference across the membrane reaches threshold, an action potential will be generated. Given this, how does the nervous system signal differences in intensity of a signal? If it's all or none, the signal either continues down the axon as an action potential or it does not. That's an on or off, zeros and ones, right? If you do any, if you are familiar with that reference, zeros and ones binary code is how we still program a lot of, um, well, it's computing language. You can do a lot with on and off, and the frequency is one way that you can do that. So frequency, so AP's action potentials per unit time. More action potentials per unit time, greater intensity. Fewer action potentials per unit time, lower intensity. You can also increase the number of action potentials and sum them together. So more action potentials summed together, higher intensity, fewer action potentials, lower intensity. So frequency, action potentials per unit time, or number of action potentials per unit time. Total number of neurons firing per unit time. Yeah, actually, okay. This number of neurons, and therefore number of action potentials. That distinction is kind of important. Um, yeah, if I simply said the number of action potentials, that's kind of the same as saying the frequency per unit time, but the number of neurons, each with its own action potential, increases the number of neuron action potentials per unit time. Okay. Frequency along a single neuron, number of neurons. Cool. That is all I have for you this week. I'm stoked that we did it in an hour. That's a lot of content. Um, get your worksheets in by Friday. You can find the recordings on the YouTube channel, PSU Learning Center YouTube channel. And take care of yourself this weekend. I will see you all next week.